Welcome to this lecture deck on fatty acid metabolism. In the next slides, we'll trace how fats are mobilized, transported, and broken down for energy, and touch on how this connects to ketone production, regulation, and disease. Think of this as a roadmap that links biochemical steps to whole-body energy balance and clinical consequences. This slide shows how fatty acids move from blood into cells and into mitochondria for oxidation. Free fatty acids travel bound to albumin, are activated to acyl-CoA in the cytosol, and then use the carnitine shuttle, CPT1 and CPT2, to cross mitochondrial membranes, a step tightly regulated to commit acyl chains to oxidative fate. Beta oxidation proceeds as a repeating four-step cycle that shortens fatty acids by 2 carbons each turn. First, FAD-dependent dehydrogenation, then hydration, NAD-dependent oxidation, and finally, thiolysis, release acetyl-CoA. Each cycle generates FADH2, NADH, and 1-acetyl-CoA, so the pathway is efficient and designed to continuously feed the citric acid cycle and electron transport chain. Breaking down palmitate produces many reducing equivalents and acetyl units, with a total ATP yield on the order of 100 after the ETC, illustrating why fats are such dense fuels. Odd-chain fatty acids end in propionyl-CoA, which is converted by biotin and vitamin B12-dependent steps into succinyl-CoA for gluconeogenesis. And defects in these steps can cause serious metabolic and neurological problems. The carnitine palmitoyltransferase system defines a major regulatory checkpoint, with CPT1 acting as the rate-limiting gate on the outer mitochondrial membrane. Malonyl-CoA inhibits CPT1, linking fatty acid synthesis and oxidation, so cells avoid burning fat when building it, and allowing hormones and nutrient signals to direct fuel choice between fats and glucose. Cells adapt fatty acid oxidation through rapid signaling and slower transcriptional programs. Energy stress activates AMPK, which lowers malonyl-CoA and relieves CPT1 inhibition to boost uptake and oxidation, while PPAR-alpha drives expression of transport and oxidation genes during fasting or high-fat intake, together enabling metabolic flexibility in response to diet and energy demand. Very long and branched fatty acids are initially handled in peroxisomes, where acyl-CoA oxidase and associated enzymes shorten chains and produce hydrogen peroxide as a byproduct. The resulting medium-chain acyl-CoAs are then shuttled to mitochondria for complete oxidation, demonstrating a division of labor between organelles for effective lipid catabolism. When beta oxidation capacity is exceeded or impaired, omega oxidation provides an auxiliary route via cytochrome P450 hydroxylation to form dicarboxylic acids. This pathway becomes prominent in diabetic ketoacidosis or inherited beta oxidation defects, producing metabolites that can be detected clinically and reflecting how alternative routes compensate when the main pathway is stressed. Inherited defects in fatty acid oxidation present with distinct clinical patterns. MKAD deficiency commonly causes fasting hypoglycemia and accumulation of medium-chain metabolites. VLKD deficiency leads to cardiomyopathy and muscle energy failure with long chains, and LCHAD deficiency affects liver and heart with long-chain blocks, highlighting the vulnerability of high-energy tissues to these enzyme defects. Excess fatty acids can overwhelm mitochondrial capacity, leading to accumulation of diacylglycerol and ceramides that activate PKC and inflammatory kinases, which then blunt insulin signaling. The result is ectopic lipid deposition in liver and muscle, and a path toward metabolic syndrome and type 2 diabetes, linking lipid overload to systemic insulin resistance. This slide integrates core pathways and control points. Fatty acids are activated to acyl-CoA. Entry into mitochondria is CPT1 regulated, and mitochondrial beta oxidation fuels ATP production, while AMPK and PPARs control activity and gene expression. Disruption of these nodes produces hypoglycemia, cardiomyopathy, or insulin resistance, underscoring the clinical significance of efficient fatty acid metabolism. Mastering fatty acid oxidation has wide implications. Efficient oxidation supports ketogenesis as an alternate brain fuel, supplies acetyl-CoA for cholesterol and steroid precursors, and enables integrated fuel switching between tissues. Understanding these processes informs nutritional guidance, therapeutic strategies for obesity and diabetes, and management of inherited metabolic disorders to optimize energy economy and metabolic health.